Thank you, Louise, and thank you for the invitation. I wasn't going to refer to just how long this project has been going on for. So yes, I've been asked to just give a kind of progress report of where we are with this activity of this expert group. So this is a systematic review of plant-based ingredients and their effects on cognitive performance. And the reason that we're interested in plants is firstly that they contain a number of components with the potential to modulate cognition. And this is important because, as we heard this morning, in terms of pharmaceutical drugs that are out there to combat cognitive decline, they are lacking. So we need some alternatives, and plants are potentially a good alternative because they don't have the side effects that we see with pharmaceutical drugs. They have the potential to actually be more beneficial. And in terms of just as a food source for nutrients, they also have the benefit of being more sustainable than non-plant-based foods. We also know from epidemiological studies that there is an association between plant ingredients and cognitive decline. And there's also data from randomized controlled trials to back up those epidemiological associations. But what's lacking here is a systematic review of the evidence for all of these plant-based ingredients and their effects on cognitive performance. So that was the purpose of this setting up this ILSI expert group. So the plan here is to summarize and evaluate current evidence in terms of key plant-based ingredients and their effects on cognition, but we're specifically focusing on randomized controlled trials. And I guess the ultimate goal here is to highlight the most effective ingredients, but then also to provide some guidance. So I'm sure it wouldn't be a surprise to say at this point that some of the findings are quite mixed and there are inconsistencies here. So ultimately, we would like to be able to provide some guidance on methodology so that we can reduce these inconsistencies in the future. So here we have a list of the plant ingredients that ended up being included in this review. So we've got 8 broad categories here. We came up with this list. I guess each of the experts came with their own expertise, and everybody suggested the ingredients that they thought showed the most promise. And then we reached a consensus on this list. So we began with amino acids. So amino acids are involved in the synthesis of protein and other biomolecules, and they're typically studied as precursors for neurotransmitters. Then we have carotenoids, so these are organic pigments in plants and algae. Typically, you would probably think of them as giving colour to things like carrots. So they give kind of orange and red colour to plants. They're largely studied in relation to eye health, but there are studies out there that look at this in terms of cognitive function. And then we have micronutrients. So here we're focused on vitamins. So vitamins are important because of their involvement in energy metabolism in neurons. They're also involved in neurotransmitter synthesis and receptor binding. Nitrate is typically studied in its beetroot form. So nitrate, we know, is it shows increases in blood flow, reduces blood pressure. So here we're working on the basis that what's good for the cardiovascular system is also going to be good for the brain. And then omega-3s, so we're all familiar with the benefits of omega-3s in terms of their role in the brain and their importance to cognitive development. But here we're focusing on plant-based sources. So we're all familiar with this in terms of a fish source, but we wanted to look at the effects from plants. And then similarly, we have phospholipids. So again, these are very important in the brain in terms of making up cell membranes, and they're involved in mitochondrial function and synaptic plasticity. And then we have polyphenols. So these are secondary plant metabolites that are broken down into phenolic acids, stilbenes, and flavonoids. Again, we know that these are involved in enhancing processes in the brain, so they're involved in signaling pathways. they can reduce neuroinflammation and also, again, have effects on blood flow. So we know that they increase cerebral blood flow. And then finally, we had prebiotics. So prebiotics being those plant ingredients that can increase the growth of healthy gut bacteria. And obviously, we've heard a lot at this meeting about the importance of a healthy gut for a healthy brain. We also had some ingredients that we had wanted to include but ended up having to exclude. So we had a botanicals category. So this was herbal extracts such as ginseng. In this case, the number of references that were returned was just too high. So there was a large number of references and a lot of them were eligible. So this review is big enough as it is without having the botanicals in there. Too large for inclusion here, but there is a plan to have a separate systematic review just of the botanicals. And then similarly for caffeine, we had far too many references to include, but in the case of caffeine, we decided that there was already enough information out there. So there are already reviews on caffeine. We know that there are robust effects of caffeine, that they are well established. And then in terms of methylxanthines as a group and also individual methylxanthines, theobromine and thiocylline, we had planned to include them, but no studies met the inclusion criteria. So if we just look at what the search criteria was. So as I said, we were focusing specifically on randomized controlled trials. We're looking at plant-based ingredients and cognition. So we searched Embase and PubMed, and as I'm sure you can imagine, there were many, many, many search terms for this. So I won't show you all of those. And what we were focusing on here was English language papers that were published in peer-reviewed journals, so we didn't include conference abstracts. And we were focused on healthy adults, so they needed to be not diagnosed with any disease. So we did include people who were from undernourished populations and older adults, as long as they weren't diagnosed with a disease. And we also included studies if it was possible to differentiate the effects in the healthy participants from any participants who were included with a disease. In terms of the study type, so as I said, there needed to be randomized controlled trials. The plant-based ingredient needed to be administered for any duration, so it could be a single dose or it could be a repeated dose for any length of time, and also in any form. So it could be a powder, a pill, a drink, a food. And then in terms of the outcome measures, they just needed to include at least one standardized measure of cognitive performance. So we were also interested in whether there were effects on mood or imaging, but they needed to have this one standardized measure of cognitive performance to be included. And so you can see here the Prisma diagram. So we started off with around 20,000 results were returned. Once we'd removed the duplicates from that, we still had 16,500 references that we needed to look at. So here we were just screening the title and the abstract of 16,500 papers. Once we'd done that, we ended up with 676 that we needed to then look at the full papers and assess their eligibility. And finally, we ended up with 171 papers that were included in the final systematic review. So I'm just going to briefly talk through the findings from this. I'm sure you can imagine it's quite a big project, so I won't go into too much detail. But starting with the amino acids, so here, aside from the ones that I've got in bold, these other amino acids, there are papers where there's one or two papers on each of these, either in isolation or one or two of them together. So not enough evidence to even draw any brief conclusions on. And then if we look at isoleucine, leucine, and valine, so these are looked at together as branched chain amino acids. So in this case, there were 4 studies. So again, not a great number of studies, but 4 studies that were all acute and all looked at the effects on cognition in conjunction with exercise. So the purpose of the exercise is just to create a state of fatigue. The exercise increases tryptophan, and that leads to this fatigue. And in this case, 3 of the studies out of the 4 showed a significant benefit, and actually the 4th study also showed a marginal improvement, which didn't quite reach significance. And it was to Stroop, which has also been shown to be positively affected in other studies. So some quite promising evidence here, but only 4 studies. And then if we move on to tyrosine. So tyrosine is a precursor to dopamine. So here we've got 14 studies, and again, this is in conjunction with a modelled deficit. So this is either environmental stressors, so it could be a heat stress, or it could be that they're performing at altitude or in sleep deprivation. So if we create this kind of deficit state, then the thinking is that it's more easy to find effects. And in 11 of these studies, they showed positive effects, and particularly in the face of these well-known stressors, and also on tasks that are more cognitively demanding, with one exception. So there was one study that showed impaired cognitive flexibility in high cognitive demand, and that is unexpected because in other studies they've used that cognitive demand as a stressor. And then tryptophan, so tryptophan being a precursor to serotonin. So here there are 7 studies. 4 of them show negative effects and 1 of them shows a positive effect. So the negative effects are expected because of the fact that this relates to serotonin. So mixed effects with these large neutral amino acids depending on which neurotransmitter they are a precursor for. And then L-theanine. So L-theanine is an amino acid that we find in green tea. So this is a non-protein amino acid. Here there are 11 studies in isolation, and this is mixed. So some of them are negative and some of them are positive. The effects tend to be on attention or speed. So L-theanine is typically thought of as a relaxant, and it does tend to show those effects in that it tends to slow people down. But a lot of studies have also looked at this in combination with caffeine. So obviously green tea contains caffeine and L-theanine, so it makes sense to look at their effects in synergy. And it also makes sense in terms of the fact that caffeine's a stimulant and L-theanine's a relaxant, so potentially putting them together will create this synergistic effect. And here 10 studies show positive effects. It's difficult to dissociate the effects of caffeine from the effects of L-theanine. So only 7 of the studies allow us to ascertain the contribution of L-theanine, and in 3 of those, the effects of caffeine were enhanced with L-theanine, but in 1 they were attenuated. And it's also important to note that the doses that they use here are very high, so they don't really represent what we see in tea. This study that showed the attenuation actually used a slightly lower dose. It's possible that we might see completely different effects with different doses, more realistic doses of what's in our diet. So some mixed findings for amino acids. They are a difficult group of compounds to study, but all of these studies are acute, and quite often they're looking at effects in the face of some kind of challenge. If we move on to the carotenoids, so here we've got 7 studies. They're all chronic. They all show positive effects. So looking at a range of ages, they tend to focus on zeaxanthine or lutein or a combination of the 2. And as I said, they all show positive effects. Beta-carotene has also been looked at in 1 study, and here they showed an effect at 3-year duration and up to 20 years, but they didn't show any effects after 1 year. So all of these studies are chronic, so supplementing from a minimum of 4 months and up to 20 years. And they also tend to use consistent doses. So this is quite promising, but only 7 studies. And then moving on to the micronutrients, so the antioxidant vitamins, there are 8 studies here. None of them show a positive effect. So whether we're looking at vitamin C in isolation or whether we're looking at all of the antioxidant vitamins together, no positive effects. Actually 1 negative effect of vitamin C on psychomotor coordination. And then similarly for vitamin D, no positive effects were shown in the studies that were included here. So irrespective of whether it was following 6 weeks or up to 7.8 years. So often people will will put forward the fact that vitamin D studies are not long enough to explain a lack of findings. But in this case, 7.8 years, you would expect to be long enough. And then the B vitamins are a little bit difficult to really draw any conclusions on. So there are individual studies of individual vitamins. So one study of thiamine B1 and one study of B3, both showing positive effects. More studies looking at B6, B12 and folic acid. So they're involved in the homocysteine metabolism. So as we heard this morning, these studies tend to look at these in combination. In this case, it's a bit mixed. So some studies show improvements. The one study of B12 showed a negative effect. And they quite often do look at this in participants who have high homocysteine. And then finally, in the micronutrient section, we have 3 studies of antioxidants mixed with other minerals. In this case, 2 of those showed positive effects, and also 3 studies of B vitamins complexed with other vitamins and minerals. And in this case, 2 out of 3 showed a positive effect. And then if we move on to nitrate, so as I said, nitrate is usually administered in beetroot. In this case, there are 8 studies. 3 of them showed a positive effect, but one of the studies that showed a positive effect also showed a negative effect. So I think based on what we know so far from nitrate, I wouldn't particularly say that this is a cognitive enhancer based on what we know. These studies do tend to be short, so they're either acute or up to 7 days. So obviously it might be different if we look at this over a longer period. And then omega-3, so this is plant-based DHA from algal sources. 2 studies here met the criteria. One of them showed a positive effect in older adults after 24 weeks, but the other one showed a negative effect. And the authors suggest that this might be due to the short duration and also the low dose. But they also use quite an unusual measure. So they're measuring forgetting. This is where they've subtracted people's delayed recall score from their immediate recall score. And it's not a standard measure, and it could potentially be impacted by effects on immediate recall that are non-significant and potentially in favour of the omega-3. I don't think we can really draw any conclusions at this point. Okay, and then on to phospholipids. So these are usually lecithin derived, and they're enhanced with either phosphatidylserine, phosphatidylcholine, or phosphatidic acid, or a combination of them. And here we've got 6 studies. 2 of them show positive effects, and 1 of them showed a negative effect. Again, the authors point to the short duration and the low dose for the negative effect. The duration seems an unlikely explanation because we do see effects acutely. So this is just, as I say, one of those inconsistencies that we need to try to better understand. And then polyphenols. So I've split this into flavonoids. So the flavonoids are usually from fruit juices or whole fruits. There are 11 studies here. 8 showed positive effects, and this is shown acutely and up to 12 weeks, a range of tasks and a range of ages. And flavonols tend to be measured in cocoa flavonols. Here we've got 7 studies showing positive effects, again, acutely and up to 12 weeks, with a range of ages and a range of tasks. In the 5 studies that didn't show a positive effect on cognition, they did show increased brain activation or improved mood. And then isoflavones are typically taken in the form of soy. Here we've got 15 studies, 8 showed a positive effect. Because of the fact that isoflavone is a phytoestrogen, these studies tend to focus on postmenopausal women. But when we look at the studies that show positive effects, although 5 of them are in postmenopausal women, 1 of them is in males and females, and 1 of them is in men only. So this isn't a woman-only effect, and affects on a range of tasks. And then resveratrol, we have 5 studies here. 2 showed positive effects on episodic memory when administered chronically to middle-aged and older adults. The other 3 studies, 2 of them were acute, and one of them was a short-term chronic study in healthy young adults, and they didn't show any effects on cognitive performance, but they did show increases in cerebral blood flow. And then finally, prebiotics, we only have one study here. It showed a positive effect on episodic memory, but this effect was shown at 2 hours. So this, although we have categorized it as a prebiotic effect, it's not likely to be a prebiotic effect at 2 hours. So it may relate to lipid metabolism or glycemic control. Okay, so that was a very kind of low-level summary of the findings so far. What we need to do now is put our heads together and try and consolidate what this all means in terms of what are the most effective ingredients. Tyrosine, it shows beneficial effects, but they're only acute, so we don't know what this means in terms of habitual intake. We can't make any conclusions about anything to do with cognitive decline based on these. Carotenoids seem to show some promise, but only 7 studies. The B vitamins complexed with other minerals might be beneficial. It's a bit of a mixed bag, and one of the problems here is that people tend not to measure the vitamin status of their participants. So giving people vitamins if they have optimal status might not show benefits. Polyphenols have attracted the most research. It's fairly positive, but as I said, it's not always consistent, so there are conflicting findings there. And that is something else that we need to look at. So the next step after that is to try and provide some guidance on why these inconsistencies come about and how we can reduce them. So dose and duration are obvious things to look at, but also the form and the food matrix. So isoflavones in soy might be different to isoflavones in red clover. Also, domains and tasks affected. The tasks affected across different studies quite often differ. So even in the carotenoids, some of the studies use the same tasks and show benefits to different tasks in different studies. And then most importantly are the participant characteristics. We really need to think about what affects, how they differ in terms of age. Sex is very, very seldom considered in any of these studies. And then most importantly, I think nutritional status. We quite often get healthy, wealthy people in who have very high cognitive function. And it amazes me that we ever show any benefits because they are at ceiling to begin with. Okay, and just a final word about the contributors. So obviously this is a big project, lots of people contributed. Special mention to Louise Dye, who has kind of been the driving force here and has the job of editing this paper. Claire Champ ran all of the searches. Lucy Goetz was overseeing this from Ilse's point of view, so she was the one herding the cats. And also a final special mention to Keith Wesness, who was the initial co-chair of this project and sadly passed away 2 years ago. And thank you for listening.